Hello my dear students, welcome back to Biology at Priyajit. In today's class, we are going to discuss one of the most fundamental and important topics in microbiology, that is bacterial growth and nutrition. Now, when we hear the word growth, we may immediately think about an increase in the size of an organism, but in microbiology, the concept of growth is slightly different. We will understand what exactly microbial growth means, how bacteria multiply, and how their population changes with time. Then we will discuss the bacterial growth curve, including the lag phase, log phase, stationary phase, and death phase. We will also understand generation time, growth kinetics, growth factors, and after that we will move into different types of microbial cultures: batch culture, continuous culture, synchronous culture, and enrichment culture. We will also discuss the interesting phenomena of Diauxic growths. Then comes an extremely important area, that is control of microbial growth, including disinfectants, antiseptics, and chemotherapeutic agents, along with their major modes of action. And finally, we will discuss bacterial nutrition, popularly autotrophic, heterotrophy, and mixotrophy. So let's begin. What is microbial growth? Let us first understand the meaning of microbial growth. Microbial growth refers to an increase in the number of cells or cellular mass of a particular microbial population with time. For bacteria, this growth is mainly associated with an increase in cell number because bacteria generally reproduce by cell division. A bacterial cell may become larger before it divides, but when microbiologists talk about bacterial growth, they generally mean growth of the population rather than simply an increase in the size of an individual cell. Most bacteria reproduce by a process called binary fission. The sequence is quite simple, that is DNA replication. It promotes chromosome segregation, then cell elongation, septum formation, separation into daughter cells. So one bacterial cell becomes two, those two become four, four become eight, 8 becomes 16, and the sequence continue 1, 2, 4, 8, 16, 32, and 64, and so on. This is why bacterial populations can increase extraordinarily rapidly when suitable nutrients and environmental conditions are available. Bacterial growth curve. Now suppose we inoculate bacteria into a suitable liquid culture medium. Do the bacteria continue multiplying at the same rate forever? Obviously no. In a closed or batch culture, nutrients gradually become depleted and metabolic waste products accumulate. As a result, the bacterial population passes through a characteristic pattern known as the microbial growth curve. A typical bacterial growth curve consists of four major phases. The first one is the lag phase, then log phase, then log phase or exponential phase, then stationary phase, then death or decline phase. Let us understand each phase carefully. So firstly we take the bacterial sample, then we culture it in nutrient broth, then incubate it at different time interval, then determine the number of cells in different time interval by spectrophotometer, and then growth curve is obtained. Lag phase. Immediately after bacteria are transferred into a fresh culture medium, there may be little or no increase in the number of cells. This period is called as lag period or lag phase. But please remember one important thing: lag phase doesn't mean that bacteria are inactive. In fact, the cells are extremely busy preparing themselves for multiplication. So during this phase, bacteria adapt to the new environment, synthesize enzymes and proteins, repair damaged cellular components, increase in cell size, synthesize RNA and DNA, and prepare for active cell division. So although we do not observe a significant increase in cell number, the cells are metabolically active. The duration of the lag phase depends on several factors. For example, age of the inoculum, physiological condition of the cells, composition of the bacterial medium, temperature, pH, availability of nutrients, and size of the inoculum. For example, if bacteria are transferred from a nutrient rich medium to a chemically different medium, the lag phase may become longer because the cells need time to synthesize new enzymes required for utilizing the new nutrients. So remember, lag phase is the adaptation and preparation of the bacteria. Second one is the log phase or exponential phase. Once the cells have adapted to their environment, they enter the log phase, also called the exponential phase. This is the phase in which bacteria divide at a relatively constant and maximum rate under the prevailing conditions. The population increases exponentially: 1 to 4, 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, then 64, then 128, and so on. During this phase, the cells are highly metabolically active, actively synthesizing proteins and nucleic acids, relatively uniform physiologically and dividing rapidly. Therefore, the log phase is particularly useful for studying bacterial physiology and biochemical activities. So it is highly important in industrial microbiology because many organisms actively produce primary metabolites during active growth. So generation time, which is represented by G, is the time required for a microbial population to double. So N0 is the initial number of cells, Nt is the number of cells after time t, and small n is the number of generations. So Nt is equal to N0 into 2 to the power n. Therefore small n will be log nt by n0 by log by 2. So growth and rate constant, another important term is the growth rate constant represented by K. It refers to the number of generations occurring per unit time. Therefore K is equal to n by T. Since generation time is G is equal to T by N, we can also write K is equal to 1 by G. So microorganism with a shorter generation time has a higher growth rate. Stationary phase. Can bacteria continue dividing exponentially forever? No. Eventually the culture enters the stationary phase. During this phase, the rate of cell division becomes approximately equal to the rate of cell death. Therefore the total number of viable cells remains relatively constant. Why does this happen? Because several factors are responsible, such as depletion of nutrients, limitation of oxygen, accumulation of toxic metabolic products, changes in pH, and limitation of available resources. Interestingly, the stationary phase is not simply a period when bacteria stop functioning. Bacteria may undergo significant physiological processes. Some bacteria may produce secondary metabolites, develop stress resistance, form endospores, alter their morphology, and activate starvation survival mechanism. Therefore, stationary phase is equal to survival and adaptation under stress. Death or decline phase. Finally, if environmental conditions continue to deteriorate, the culture enters the death or decline phase. Here, the rate of cell division becomes greater than the rate of new cell formation. So as a result, the number of viable cells decreases. Major causes include severe nutrient depletion, accumulation of toxic substances, unfavorable pH, oxygen depletion, and prolonged environmental stress. One important point for examinations is that total cell number and viable cell number are not necessarily the same. Dead cells may still physically remain in the culture and may be detected even though they are no longer capable of reproduction. So when we talk about the death phase of a bacterial growth curve, we are particularly concerned with the decline in viable cells. Microbial growth kinetics. Now let us move to microbial growth kinetics, which deals with the mathematical description of microbial growth and the factors influencing the growth rate. During exponential growth, the bacterial population can be expressed as Nt is equal to N0 into e to the power Nt, where Nt is the population at time t, N0 is the initial population, mu is the specific growth rate, and t is time. So the specific growth rate mu is equal to 1 by n into dn by dt. So the relationship between specific growth rate and generation time is G is equal to 0.693 by mu. Therefore, a high specific growth rate means a short generation time, and the growth rate can be influenced by temperature, pH, nutrient concentration, oxygen availability, water activity, osmotic conditions, toxic substances, and the metabolic state of the organism. Growth factors. Now, what exactly does a microorganism needs for growth? Apart from major nutrients, some microorganisms require certain organic compounds in a very small quantities. They are called growth factors. Growth factors is an organic compound required for growth that a microorganism cannot synthesize sufficiently by itself. Examples include vitamins, amino acids, purine, pyrimidine, and certain fatty acids or related compounds. For example, vitamins may function as components of coenzymes or participate in metabolic pathways. Amino acids are required for protein synthesis and other biosynthetic reactions. Purine and pyrimidines are essential for nucleotide synthesis and therefore for the formation of DNA and RNA. Based on nutritional independence, microorganisms can be broadly described as prototrophs which can synthesize all essential growth requirements from relatively simple nutrients. Auxotrophs require one or more specific nutrients or growth factors from the external environment. For example, if a bacterial mutant cannot synthesize a particular amino acid, that amino acid must be supplied in the culture medium. Types of bacterial culture. First one is a batch culture, which is essentially occurs within a closed system. A fixed amount of culture medium is inoculated with microorganisms and no fresh medium is continuously added and no culture is continuously removed. Initially, nutrients are available, but as bacteria growth, nutrients decrease, waste products increase, environmental conditions change. Therefore, the culture is normally passes through lag, log, stationary, and death phase. Advantages of batch culture: it is simply economical, useful for laboratory studies and suitable for more fermentation processes. Limitation: however, nutrients become depleted, toxic metabolites accumulate, physiological conditions change continuously, and it becomes difficult to maintain bacteria indefinitely in the exponential phase. Continuous culture. Now imagine that we want to keep bacteria growing for a much longer period under relatively controlled conditions. For this purpose we use continuous culture. In a continuous culture, fresh medium continuously enters the growth vessel while approximately the same volume of culture is continuously removed. The two important systems are chemostat and turbidostat. Chemostat controls microbial growth by limiting the concentration of a particular nutrient. The growth rate is influenced by the dilution rate and concentration of the limiting nutrient. Turbidostat, which maintains a relatively constant cell density or turbidity. When cell density rises above a set level, fresh medium is added and equivalent amount of culture is removed. Batch culture versus continuous culture. Here is a comparison between the two process of cultures. Then we go for synchronous culture, where all cells are not necessarily at exactly the same stage of the cell cycle. A synchronous culture is one in which a large proportion of cells proceed through the cell cycle at approximately the same time. This is extremely useful for studying DNA replication, chromosome segregation, cell division, cell cycle regulation, cellular morphology, and the timing of macromolecular synthesis. Synchronization can be achieved by methods such as physical separation according to cell size, selective filtration, density based methods, and controlled environmental shifts. However, synchronization is often temporary because cells gradually lose synchrony as the culture continues to grow. Enrichment culture. Now comes another very important concept. Enrichment culture. Suppose we have a mixed microbial population and we want to isolate a particular microorganism. How can we encourage our organism of interest to grow while discouraging the others? The answer is enrichment culture, which is designed to selectively favor the growth of a particular microorganism or group of microorganisms from a mixed population. We can manipulate carbon source, nitrogen source, pH, temperature, oxygen availability, salinity, isolate a microorganism capable of using an unusual carbon source. So then the technique is widely used in environmental microbiology, soil microbiology, microbial ecology, industrial microbiology, and the isolation of specific specialized metabolically specialized microorganisms. Diauxic growth. Now let us discuss a fascinating phenomena called diauxic growth. The diauxic essentially indicates a two-phase growth curves. Di means two, or ox means growth. So it occurs when microorganisms are provided with two different carbon sources, particularly when one is preferred over the other. The classical example is E. coli growing in a medium containing glucose and lactose. What happens? First, the bacteria uses glucose because it is the preferred carbon source. So we see the first exponential growth phase. When glucose becomes depleted, the bacterial growth temporarily slows. This short period is called the lag period between the two growth phases. During this time, the cells adapt metabolically and synthesize the enzymes required for lactose utilization. When growth begins again, producing the second exponential phase. So the pattern is first exponential phase, short lag, and then second exponential phase. This is called Diauxic growth. Role of catabolite repression. Diauxic growth is closely related to catabolite repression. When glucose is abundant, the bacterium does not waste energy synthesizing enzymes needed to metabolize less preferred carbon sources. When glucose concentration falls, intracellular cyclic AMP increases. Cyclic AMP then binds to catabolite activator protein, or also called CAP or CRP. The CAP complex helps activate genes involved in the utilization of alternative carbon source such as lactose. At the same time, the regulation of the lactose system involves the lac repression. Thus, bacterial cells efficiently prioritize their carbon sources and avoid unnecessary expenditure of nature and expenditure of energy. So this is beautiful example of metabolic regulation in bacteria. Control of microbial growth. Now we come to another extremely important topic, control of microbial growth. Why do we need to control microorganism? Because microorganism may cause infectious disease, food spoilage, contamination of laboratory materials, and problems in industrial processes. So microbial control can involve physical method, chemical method, and antimicrobial drugs. Before discussing disinfectants and antiseptics, let us distinguish four important terms. Sterilization, which means the complete elimination or destruction of all forms of microbial life, including bacterial endospores. Disinfection, which means destruction or removal of many or all pathogenic microorganisms from inanimate objects. However, disinfection does not necessarily destroy bacterial endospores. Antisepsis means the application of antimicrobial chemical agents to living tissues to reduce or inhibit microorganisms. Sanitization means reducing microbial populations to level considered safe according to public health standards. These four terms should not be confused. Disinfectants. A disinfectant is a chemical agent primarily used on non-living surfaces to reduce or destroy microorganisms. Major groups include phenolic compounds, halogens, alcohols, aldehydes, oxidizing agents, and quaternary ammonium compounds. Biguanides and heavy metals. Let us briefly understand their mechanisms. Phenolic compounds, they mainly act by disrupting cell membrane, denaturing protein, and damaging cellular components. Halogens, there are two important examples are chlorine and iodine. Chlorine is widely used in drinking water treatment, swimming pool, and surface disinfection. Its antimicrobial activity involves relative chlorine species, particularly hypochlorous acid, which damages proteins, membranes, and other essential cellular components. Iodine acts through oxidation and interaction with proteins and cellular structures. Iodophores release iodine gradually and are widely used as antiseptic and disinfectant preparation. Alcohols example include ethanol, isopropanol. They mainly denature proteins and disrupt lipid membranes. Alcohol act rapidly against many vegetative bacteria and enveloped viruses, but they are not reliably sporicidal agents. Aldehydes are example include formaldehyde, glutaraldehyde. They act mainly by modifying and cross-linking proteins and nucleic acids. Oxidizing agent examples include hydrogen peroxide, pre-acetic acid, ozone. They cause oxidative damage to proteins, membranes, lipids, and nucleic acids. Quaternary ammonium compounds, these are cationic detergents. They primarily disrupt cytoplasmic membranes, alter membrane permeability, and cause leakage of cellular contents. Biguanides, an important example is chlorhexidine. It interacts with microbial membranes, increases membrane permeability, and causes leakage of cellular constituents. Disinfectant versus antiseptic. The easiest way to remember the difference is disinfectant to dead or non-living surfaces, antiseptic or living tissues. Disinfectant is primarily intended for inanimate surface, whereas antiseptic is formulated to be sufficiently safe for application to living tissue at an inappropriate concentration. Examples of antiseptics include alcohol preparation, iodine preparation, chlorhexidine, and certain oxidizing agents. One important point, the same chemical may have several applications depending on its concentration, formulation, and the intended use. So this is the differences showing the antiseptic and disinfectant. Chemotherapeutic agents. Now we move from environmental control to antimicrobial therapy. A chemotherapeutic agent is a chemical substance used to treat microbial infection within a host. Unlike disinfectants, these agents need to act inside the body. Therefore, an ideal antimicrobial drug should show selective toxicity. What is selective toxicity? It means the antimicrobial agent should inhibit or kill the microorganism while causing minimal damage to the host. This is one of the fundamental principles of antimicrobial chemotherapy. So major targets of antimicrobial agents, the targets are cell wall synthesis, cell membrane protein synthesis, DNA synthesis, RNA synthesis, and foliate metabolism and other metabolic pathways. So these are the different targets of antimicrobial agents. Cell wall synthesis. The bacterial cell wall contains peptidoglycan which is absent from human cells. Therefore, bacterial cell wall synthesis provides an excellent target for selective toxicity. Beta-lactam antibiotics examples include penicillin, cephalosporins, carbapenems, monobactams, etc. They target penicillin binding proteins including transpeptidases and interfere with peptidoglycan cross-linking. The weakened cell wall may ultimately lead to osmotic lysis, particularly in bacteria actively growing bacteria. Vancomycin, which is a glycopeptide antibiotic, it binds a terminal peptide sequence of peptidoglycan precursors and interfere with cell wall synthesis. It is particularly important in the treatment of gram-positive bacterial infections. Protein synthesis. Bacterial ribosomes are 70S, consisting of 30S small subunit and 50S large subunit. Bacterial ribosomes differ structurally from eukaryotic cytoplasmic ribosomes. They are excellent targets for antimicrobial agents. 30S inhibitors, aminoglycosides such as gentamicin and streptomycin, interfere with the 30S subunit and cause errors in translation. Tetracyclines interfere with amino acyl tRNA binding and inhibit protein synthesis. 50S inhibitors, macrolides such as erythromycin, bind to the 50S subunit and interfere with translocation. Chloramphenicol inhibits the peptidyl transferase reaction. Clindamycin binds to the 50S subunit and inhibits protein synthesis. So for examination purpose, 30S is the aminoglycosides and tetracyclines, 50S is the macrolides, chloramphenicol and clindamycin. Inhibitors of nucleic acid synthesis, fluoroquinolones. Examples include ciprofloxacin, levofloxacin. They target bacterial enzymes involved in DNA replication, particularly DNA guidase and topoisomerase 4. Therefore, DNA replication and chromosome processing are disrupted. Rifamycin inhibits bacterial DNA-dependent RNA polymerases. Therefore, it blocks transcription and consequently RNA synthesis. Antimetabolites resemble normal metabolic substrates or intermediates and they interfere with essential metabolic reactions. A classic example is inhibition of folate synthesis. Sulfonamides resemble PABA, para-aminobenzoic acid, which require PABA for folate synthesis. Sulfonamides inhibit the enzyme dihydroptorate synthase and interfere with folate biosynthesis. Trimethoprim inhibits dihydrofolate reductase. When sulfonamides and trimethoprim are combined, they can produce sequential inhibition of folate metabolism. Why this pathway is good for antimicrobial target? Because humans primarily obtain folate from their diet rather than synthesizing it through the bacterial pathway. So therefore, bacterial folate synthesis provides an excellent target for selective toxicity. Bactericidal and bacteriostatic agent. Bactericidal agents kill bacteria. Examples include beta-lactam, aminoglycosides, fluoroquinolones, etc. Bacteriostatic agents include which inhibit bacterial growth and multiplication. Examples include tetracycline, macrolides, sulfonamides, etc. This is the difference in between bactericidal and bacteriostatic agents. Nutrition of bacteria. Now we come to the final major part of today's lecture, bacterial nutrition. Microorganisms require nutrients to obtain energy, synthesize cellular components, growth, reproduce and maintain cellular functions. Based on their nutritional strategies, microorganisms can be broadly discussed as autotrophs, heterotrophs and mixotrophs. Autotrophs use carbon dioxide as their principal or sole carbon source for synthesizing organic cellular compounds. But where does their energy come from? It may come from light or oxidation of inorganic substances. Therefore, autotrophs can be divided into two major categories: photoautotroph and chemoautotrophs. Photoautotrophs obtain energy from light and carbon, so carbon dioxide, so they use energy to drive carbon fixation. Examples include cyanobacteria, purple sulfur bacteria, green sulfur bacteria. So remember, photoautotroph is equal to light energy plus carbon dioxide carbon source. Chemoautotrophs obtain energy, oxidation of inorganic substances and carbon from carbon dioxide. Possible inorganic energy sources include ammonia, nitrite, hydrogen sulfide, enzyme, elemental sulfur, hydrogen ferrous ion. For example, nitrifying microorganisms obtain energy by oxidizing reduced nitrogen compounds. Thus, chemoautotroph means chemical energy from inorganic substances, and carbon dioxide, CO2 as carbon sources. Heterotrophy. Now let us move to heterotrophy. Heterotrophs obtain their cellular carbon primarily from preformed organic compounds. These compounds may include carbohydrates, proteins, amino acids, lipids, organic acids. Many bacteria, fungi, and protozoa are heterotrophic. Heterotrophic microorganisms may obtain organic nutrients in different ways, such as saprophytes, parasites, and symbionts. Mixotroph. Mixotroph. Finally, we have mixotrophy. Mixotrophy refers to a nutritional strategy in which organism can use more than one mode of nutrition, combining characteristics of autotrophic and heterotrophic metabolism. A mixotrophic microorganism may fix carbon dioxide under certain conditions, use organic carbon under other conditions, or sometimes use inorganic and organic carbon sources depending on the environment. This flexibility becomes particularly advantageous when environmental resources fluctuate. For example, some microorganisms, particularly certain algae and protists, can switch between photosynthetic growth or autotrophic growth and then heterotrophic growth. So depending on light availability, organic nutrient availability, environmental stress, and availability of inorganic carbon. So the easiest way to remember three nutritional strategies is autotroph, carbon dioxide is the carbon source; heterotroph, organic compounds are the carbon source; and mixotroph can use more than one nutritional strategy. So this brings us to the end of our discussion, that bacterial growth and nutrition. So we covered bacterial growth, growth kinetics, culture systems, Diauxic growth, microbial control, antimicrobial mechanisms, and bacterial nutrition. If you found this class helpful, please like, share, and subscribe for more student friendly microbiology classes. Thank you for watching. Keep learning, keep revising, and keep growing.